blatant self-promotion number one for today. Hopefully we can get this trending on Twitter. Someone announced our paper that just came out. So American Society for Virology. Look at those amazingly cool and interesting viruses, and particularly the mutant viruses that we have here. Mutant viruses from hell. It's always a good time, relatively speaking. So I just wanted to, again, toot our own horn a little bit here. Let's see, we can get this going. Hopefully everyone liked the flavivirus guest lecture. Yes, good. How many of you heard Bill Martin yesterday? Some of you in microbiology? Fun stuff? I have my difference of opinion with Dr. Martin, but we could talk about those later. But that being said, Ian's here, so we can start with our clicker questions, right? Perfect timing. So, which is the most recent flavivirus to be discovered or have been discovered is probably a better way of putting it. Hepatitis C, dengue, West Nile, yellow fever, or Zika? Process of elimination.
I may not know the answer, but I know not the answer.
10, 5. Okay, well, hopefully we'll have some that have 100% later on, but this one, pretty obvious that we don't. And in fact, it's rare that we have such a difference of opinion here. That's pretty impressive. That's very impressive indeed. So I think basically we've kind of been guessing on this one. So, really, you think? How on earth could I get that idea? So most recent flavivirus to be discovered is the one that was only discovered molecularly in terms of discovering the virus. Which one is that? Hep C. So it's hepatitis C. Zika was discovered in the 40s. This was the poor monkey on top of his tree, artificial tree, in the Zika forest. So yes, a lot of people care about Zika, but that's only because of the recent outbreaks. Yeah?
Didn't your colleague yesterday, didn't he say that Zika was discovered with a few others in that same study?
Yeah, so multiple viruses were discovered at that same time, but hepatitis C is much, much, much later. Okay, so, and I will also cover a few more of these things when we do our review already. Wow, it's almost midterm time. How did that happen? So, yeah, no, don't tell me about it. So, we feel a little better about the next question. We're getting in the mood for flaviviruses? No. So we'll start with this one instead. So which of the following is currently causing the highest number of deaths in the world? Dengue, West Nile, yellow fever, or Zika? Again, feel free to discuss this. I know you're all blown away by Bill Martin and Dr. Wiesler then, so.
It's never Zika. Which is the Zika virus? It's not Zika.
That's true. That eliminates, 25%.
I remember one of them. I don't know if it was still a big problem.
10, 5. Okay, we are definitely getting better. So the next one hopefully will be it. Now, dengue by far is really the one which is causing most disease in the world, and that's because there aren't good vaccines. There are pretty good vaccines for yellow fever. So there are sporadic outbreaks of yellow fever, but it's much, much, much lower than dengue in terms of everything else. And yeah, Zika, practically zero mortality from Zika. I think there have been about 4 or 5 cases total of actual fatality from Zika. So yes, it's A. It always seems to be A. I wonder if that's something that I forgot to, well, that's not going to give it away for the next one, will it? No, okay. So our last Zika clicker, or Zika clicker, did I say Zika clicker? Flavivirus clicker. So flavivirus replicates plasma membrane in endosomes at membranes in the cytoplasm at the nuclear membrane in the nucleus. We didn't talk about molecular stuff, did we? No, we did. That's not one of the possible answers. Oh no. 10. So we're asymptotically getting closer to 100%. So it is membranes in the cytoplasm, probably derived from endoplasmic reticulum, but this should sound really familiar because that's where the picoRNA viruses are replicating as well, and as we'll see today, the coronaviruses as well. So many of these positive strand, single strand RNA viruses are replicating at internal membranes. So, and we will talk about other examples of basically all of these as we move through the class. So there are examples of all of these. We won't talk about the endosome replication ones, but there are a couple that in fact replicate in the endosome as well. Okay, so now shift over. How many of you made it to Dr. Wiesler's seminar yesterday? Great introduction to coronaviruses. Talk a little bit more about some of the current coronavirus diseases which are circulating. Well, there's the common cold that's circulating all the time, but the thing that most people seem to care most about are these ones over here, SARS and MERS. SARS is a really pretty amazing example, as far as I'm concerned, of public health and surveillance being incredibly good and incredibly fast in terms of picking up viral diseases. MERS is, if anything, more of an issue, and certainly more people have died from MERS, I think, than from SARS. No one really thinks that much about MERS, but SARS was a much bigger deal. So we'll talk a little bit about that towards the end, but the most important aspect of the coronaviruses, again, as far as my totally biased opinion is concerned, is that these are really enormous single-stranded RNA viruses. In fact, the largest known RNA viruses are these coronaviruses. They're enveloped, again, not a huge surprise, and that was the sort of main take-home message from Dr. Wiesler's talk yesterday, is how a lot of those envelope proteins are involved in getting this very large virus genome here on the inside into the host cell. And so he's mostly talking about this virus spike protein here on the outside. A couple of key concepts that, again, Dr. Wiesler talked about, but since apparently there's a few who were there. We need to talk about them again. Many of the receptors that coronaviruses interact with are extracellular, I should say, really transmembrane proteases. And so they're proteases which reach out into the external environment and are probably important for most cells to break down some of the food, as it were, for any of these individual cells. And now the virus has taken advantage of the fact that these are these extracellular receptors and binding to them. In terms of the replication of the genome, something we haven't really talked about, or we talked about really briefly for MS2, is this concept of frame shifting. And so it's a translational change that happens while that genome is being translated. And we'll look at that in more detail. And then probably the most molecularly interesting aspect about these viruses is that instead of just one big RNA, because again, it's a positive strand RNA, it encodes everything that it really needs in that one RNA, but it's got the same problem that many of these other RNA viruses have. You need a whole bunch of some sets of proteins and not that many of some of the other proteins, particularly structural proteins you need a lot of, and the non-structural proteins you need a lot less of because they're mostly enzymatic. And how that works here in the coronaviruses, and as we'll see for a lot of the negative strand viruses as well, these things called subgenomic RNAs. So it's not the whole positive strand that gets translated all the time. It's just smaller pieces that get made. And how those smaller pieces get made we'll talk about in considerably more detail. And then the whole concept of reservoir species. I know Dr. Hirsch brought this up last time, but we'll cover this in a little bit more detail. And for the coronaviruses, particularly for MERS and SARS, it's really important to get the concept, because probably for less so for MERS, but and definitely true for SARS, is that these are viruses that don't transmit very well from person to person. And so where do those viruses come from? They come from a reservoir species, and almost definitely bats in this case. And we'll see how some of that data was brought up. So our classic outline here. Here's our friendly little virion. Actually, they're relatively large virions. Where they come from, structure, binding and entry. And then the real key here is this messenger RNA production and replication, which is really quite different with these viruses than any of the other ones that we've talked about so far. Translation, it turns out, is pretty normal, and release is pretty normal as well. So where do these coronaviruses come from? The vast majority were known to be those that were causing cold. Everyone gets well pretty easily, so who cares? And in fact, we'll look in just a second to see that there was very little work that was being done on coronaviruses until, let's see, about 2003, when we had this virus that started to infect humans. And of course, well, 2003 wasn't that long ago, so most of you hopefully remember all of this, craziness here that was going on. Now, SARS, all over the place. And it was, let's put it back up a little bit. A big deal was made about SARS as far as that was concerned. And the main reason for that is also why people got so excited about, or depressed, or lots of media about Ebola, is that high death rates from people who are infected with this. And that's the main reason that the media goes nuts with these things. When we talk about flu, I think next week, we'll see that way more people die from flu than any of these other diseases probably combined. So, but do you see much about flu in Time, Newsweek, US News and World Report? Practically nothing. So, coronavirus disease. Again, the vast majority of this was the common cold. What causes probably the rest of the common cold? They had a nice little cough up here at the front. It's appropriate. The rhinoviruses, yeah, so the picoRNA viruses. So between rhinoviruses and coronaviruses, 90+% of upper respiratory tract infections. Turns out that there's also many, many animal coronaviruses, which don't infect humans, which is really nice because it makes a nice system to study. And in fact, murine hepatitis virus is the one which has been probably best studied. And in fact, that was the one that Dr. Wiesler studied the structure thereof, although it was a whole mixture of different things that he was studying there. So, okay, who cares about coronaviruses? They infect dogs and cats and sheep and everything else. Yeah, boring. Until, of course, SARS comes along. This is, as I mentioned right at the beginning, a pretty amazing example of public health and surveillance. The first reported disease was in February of 2003, and the virus was discovered in May, 4 months later, which is pretty insane in terms of a new viral disease. So for hepatitis C, we knew about non-A, non-B hepatitis for decades, and then finally found the virus much later on. Here, it was in the process of months. There were about 1,400 cases by the following month, so spread quite quickly. One of the issues there may have been the surveillance. There probably were more examples of SARS before 2003. So what is SARS? SARS is severe acute respiratory syndrome, so not a common cold. And the main thing here, again, is a very, very severe process. We don't actually know very much about why SARS causes so much nastier disease than any of the other coronaviruses. It's a very interesting question that we haven't really picked up on and figured out what's going on there. What seems to be happening is that there's so much fluid which is building up in the lungs that there's not enough oxygen pickup. And so it seems to mostly be this amount of oxygen and lack of oxygen which is really leading to the nasty disease. Eventually, there were almost 9,000 cases and little under 10% death. Now, of those, 8 cases were in the US. That's why you want to be on the cover of all of these magazines, right? 8 cases, and nobody died. So it tells you a little bit about the media aspect about these things. And it's not just the media. We'll get to some of the rest of this later on down here. So how do you get infected by SARS? Mostly it was direct contact with animals, and this was the live animal markets in China. The original thought in terms of where it came from were the civet cats, and these are small cat-like mammals that are apparently incredibly prized. I've never eaten any, but apparently the meat is very good. And in China and a lot of other places in Southeast Asia, very often you get your meat live at one of these live markets. And so the thought was that it came from the civet cats, and the virus was actually isolated from these civet cats. Considerably later, sorry, was there a question?
Oh yeah, well you said the virus was isolated from them.
Yeah.
Was it also causing respiratory infections in?
Was it causing disease in these cats? There didn't seem to be much in the way of disease in these cats. And probably the actual reservoir, which again we'll talk a little bit more about later on, is almost definitely in the bats. And how the bats got to the cats and how those cats transmitted it is still, I think, an open question. But probably it was what people call a spillover event. So these viruses were probably circulating in the bats. There was no problem with the bats. They actually had 0 phenotype. And those then infected, in some event, the cats, which then infected the people. There have been, since 2003, over 4,000 papers written about SARS. I don't know if this is a nice approximation. So I said, the media went nuts about this, but 8,000 cases, 4,000 papers have been written about it. So that's like 1 paper for every 2 cases of SARS, which seems to me be a little extreme. Giving you a bit of an idea, how many of these scientific papers had been published before 2003, and this is the whole medical literature before 2003, there were 129 papers on coronaviruses. After SARS, now we've had 4,000 papers that have been published on this. Yeah.
Is there any way to look at, once you've isolated the virus and once you've identified it structurally, or how much structural identification do you need to go through to make a determination if you think that the virus is capable of mutating such that it could become more infectious in terms of transmission from the vector animal to humans, basically?
Okay, so I'll paraphrase your question here. We'll talk more about the whole idea of transmissibility, getting from the reservoir species into the human population and then spreading from humans to humans. There has been a lot of work on flu, all the high pathogenic flus, et cetera, avian influenza, that business. That's been pretty well studied, but the problem is we really don't know what that is. And so the experiments need to be done. And I'm kind of paraphrasing TWIV. Any of you people who listen to TWIV, you need to do the experiments because you can't predict from the sequence what's going to happen. And that seems to be the case here for a lot of these viruses as well. Dr. Wiesler talked a little bit about this yesterday in his seminar about learning about the structure. And this is the molecular structure of some of the pieces of these coronaviruses that might explain some of the pathogenesis, but pathogenesis mostly because of the escape from the immune system. But a lot of this is hand-waving in terms of trying to understand that. And then, where did SARS go? There haven't been any natural cases since 2004. There were, in fact, a couple of laboratory-acquired cases of SARS because people were working on it. But the actual outbreak was done in a little over a year, which is pretty amazing that you have this disease, it comes up, then disappears, but provides all kinds of good material for scientists to continue working on it. Because there's been laboratory-acquired infections, it was declared what's called a select agent in 2013, and basically a select agent means there's practically no way that you can work with it in your lab. It's highly controlled in terms of you can actually get then your hands, as it were, on one of these viruses. So practically all of the studies which are done in little bits and pieces of different coronaviruses, and many, many of them on these animal coronaviruses, which actually don't infect humans. So that brings us back to the reservoir species. Really concerned, you have this disease, shows up, kills a couple hundred people. Okay, this is why we're all concerned about it. But still, nonetheless, is this emerging process of disease. Where does it come from? Yeah, Angela.
Well, I mean, isn't there a potential for a pandemic if it's going from an animal to a person? And maybe it's not that transmissible between people, but if it overcomes that, the common cold is very transmissible, then there is a potential.
Right, so the question is how much to be concerned about it. And I may be brushing it off probably more than I need to. But if you look at it, it was around for a year. In that year, it was about a 10% fatality rate. And we get a chance, which we're probably not going to. MERS has even a higher fatality rate. It's about a 35% mortality rate. But the numbers there are extremely low. And probably because of that, just from an epidemiological point of view, if there was going to be massive spread, it would have happened already. And we have a pretty good idea how many mutations happen in replication here. And this is kind of getting to Ian's question a little bit earlier on. Turns out that the studies that they did on flu to look at how rapidly it could transfer and then making changes that allowed it to transfer ended up lowering the pathogenicity. So probably, and again, if you think about this from the holistic point of view, if you're a virus, again, to totally anthropomorphize, and you kill off your host and you're host dependent for replication, it's kind of a stupid way to replicate. So these highly pathogenic diseases probably are not an appropriate way for the viruses to replicate. Because if they did, the virus is basically killing itself off because it's so dependent on hosts. So that brings us to the reservoir species, because this is where the virus seems to be hanging out, and it's perfectly happy because it's not causing disease in these particular hosts. So it turns out bats, for all kinds of interesting reasons that I did talk to Dr. Wiesler about before, probably having to do with innate immunity and the fact that they're flying mammals and have very different metabolisms. They also have different immune systems. They seem to be able to deal with all kinds of viruses that for us are highly pathogenic with practically zero pathogenesis. See almost no kinds of phenotypes, no disease whatsoever in these animals, which then makes sense that the virus is dependent on the host, but this host is perfectly happy with it, and so that virus can spread actually through these particular hosts. And so this is the whole concept of the reservoir species, is that that's where most of the viruses are circulating. And occasionally there'll be this, what they call a spillover event, where it'll be the civet cats, or in the case of Ebola, actually the outbreak in West Africa that we'll also talk about, probably had a case where the bats came into contact with the humans and then they got infected. And so that's the idea of these reservoir species. And so one of the things that people noticed is that at least 50% of the bats in the Philippines had coronavirus-like proteins circulating in their blood. Bats in the US also have coronaviruses that are associated with them. There have been 0 cases in the US of any transmission or spillover event. So probably it's actually a pretty rare event, because if you think about how many viruses there are in these animal species, that we just had that, well, basically 2 cases now of that transmission, it's pretty rare that something like that happens. And usually humans in those cases are what we call a dead-end host. They can't spread the disease very well, or that disease doesn't spread very well from human to human unless they're in really close contact, and that turns out to be where we've had almost all of these cases. And so when people talk about quarantines and isolation, that seems to make the vast majority of the difference in these cases. Okay.
Human is not a dead end for SARS.
Human is not completely dead end, and so the problem with dead end is that that's a black or white kind of thing, and so you can have human-to-human transmission, but it's pretty rare that you have that human-to-human transmission, and it's usually when people are family members very closely associated with each other. And we'll talk more about that with Ebola and some of the filoviruses as well.
Yeah, I was thinking that, like, with the avian flu, I mean, it doesn't transmit that well between people, but it does.
Yeah, well, we'll talk much more about flu when we get to the flu lecture. So, yeah.
Could you use bat cells for viruses like this to develop lactase to study their mechanism?
So, question is, can you use bat cell cultures to set this up? And in fact, Dr. Ruedas from our department was talking to Dr. Wiesler about this after his seminar to try and set up some bat cell cultures to try and understand what's going on with that. May have to do with more of the innate immunity of the whole bat, so that's a little bit harder to do. So the other indication that these are the reservoir species has to do with the just sequence comparison, and not so much sequence comparison in terms of thinking about pathogenicity, but just if you think about where these viruses are likely to come from, if they've been in a set of organisms for a long period of time, when I say a long period of time, multiple replication cycles, these viruses are mutating, you're going to expect to see way more diversity of virus sequences in those reservoir organisms than you are in these sort of dead-end and not very well transmitting organisms. And so that's exactly what was seen with the SARS genomes. So SARS of human and the civet cat are basically identical to each other. People used to look in these trees. Sorry, I should maybe back up a little bit. So the idea here is the length of each of these horizontal bars represents then sequence differences. And so sequence differences between the human SARS and the civet cat SARS are identical. So that was why people thought that it was coming from the civet cats. But it turns out that you also find lots of civet cats that are not infected, and then very little diversity present in those civet cats. On the other hand, there's a bat virus, also very similar to these sequences, and then all of these other sequences are bat sequences. So clearly there's a huge amount of diversity in bat coronavirus sequences, and just in this case, there seems to have been this spillover event which has taken place. So many of these are actually even sequences that people have just found. They haven't even gone into a bat. They just found a sequence of a coronavirus. Turns out to be relatively easy. We talked about the metagenomics before. Many of these are metagenomic sequences as well. But those that have been identified as having a host, the vast majority of these are bat sequences. So huge amounts of diversity in that reservoir species, no symptoms which are going on in that particular species, and occasional cases of extremely similar viruses now in slightly different hosts. Any questions on reservoir species, et cetera?
A little bit. Is it just one of many types of vectors? Does it just refer to a living species that can carry the virus without getting infected? Or is there some difference from the vector?
Oh, so the question, I guess, here is reservoir species rather than vector. Yeah. So reservoir and vector are two slightly different things here. So vector is something that you need to transmit the virus. And so it's going usually from one organism to another. And so in the case of the flaviviruses, vast majority of them, not hepatitis C, these then have mosquito transmission. But it's probably, the mosquito is probably not the reservoir. That's probably not where most of the virus is hanging out most of the time, not causing some kind of disease. At least for yellow fever, it's probably some of the other primates which are present. Dengue is a little less obvious what the reservoir species is, but the idea of the reservoir is that's where the viruses are continually replicating and continually transmitting as well from organism to organism. Yeah?
Is it kind of like the cucumber mosaic virus and the aphids?
Yeah, so the aphids would be the vector in that case. But there doesn't seem to be a clear reservoir for those. And again, the idea of the reservoir is it's where you have this free transmission, large amounts of diversity which is there, and usually applied for animal viruses, and particularly viruses which are going to infect humans.
To have a reservoir?
You have to have a reservoir. You don't have to have a reservoir per se, and in fact, in some cases, the reservoir is humans. So polio is actually a really nice example of that. Only infects humans. So the reservoir species for polio is humans, which actually makes it possible to eliminate it. If in many cases we don't know what the reservoir species are for many, many of these different viruses. Okay, so we're clear on reservoirs now. So clear that we can get our clicker questions out and get 100%, right? We're going to do 100% here. That's the plan. The highest diversity in SARS-like coronaviruses is known in bats, beluga whale, ferrets, humans, mice. Oh, let's start. That would help.
I thought I heard something about beluga whales in there.
I'll explain the beluga whale a little later.
I'm really curious about reservoir and rabies, because rabies is fatal to every species that I know of that it resides in. So where does it possibly, where can it be in reservoirs? I mean, I think it lasts longer than bats, but they do succumb.
Doesn't have a really variable replication cycle depending on the species. Like some species can live a lot longer with it than others can.
Probably, that's probably true.
I've heard that
At least with domestic.
Yeah, and it might have to do with the levels as well. Yeah. So what viremia is just talking about last time. You know, what the virus levels are.
I actually pulled up one and just scrolled through it.
So anyone else want to vote? Are we all done? 5. Vote early, vote often. It's the last one that counts. And Ian's going to answer the wrong question just to mess me up. I know he will.
Beluga whale.
Yes. hallelujah. And A again, how did that happen? So yeah, the whole beluga whale business. This is a holdover. Some of you may have looked at some of the old exams. I used to also have problem sets. One of the problem sets was to go and find the largest RNA genome. And it turns out that there's a coronavirus that infects beluga whales, which has the largest RNA genome. So that's where the beluga whales thing comes from. Ferrets turn out to be an incredibly useful tool for studying number of virus diseases, but particularly flu. Humans, practically none, and mice, actually, it's the mouse hepatitis virus, which is the coronavirus that people work with. So let's make sure, come on, let's make sure this is right and it gets stored appropriately. So that's all I wanted to do in terms of talking about disease, at least for SARS, and maybe we'll get to MERS right at the end here. So why coronaviruses? Corona is crown, and that's because they have these really amazing spike proteins that stick out on the outside of each of the virions. And that's also known as a spike protein, and we'll talk a lot more about that a little bit later on. Classic enveloped viruses, so it's a membrane that's surrounding the nucleic acid. That nucleic acid is packaged in a helical form. And so for SARS, for most of the coronaviruses, it turns out there are a couple of those that have the nucleic acid that's packaged in an icosahedral-like structure. This nucleocapsid, and I don't think we really talked too much about nucleocapsids so far. So the idea of a nucleocapsid is it's protein plus nucleic acid inside an envelope. And in some cases, it will be a really classic icosahedrally symmetric particle. Many of these enveloped viruses, particularly the ones with the larger genomes, are packaging their nucleic acid in a helical form, which again makes a lot of sense because these are also very helical in terms of their structures. So this is easier to look at in a cartoon. The spike protein, this is really the major business end of the virion. It interacts with the receptors. In the case of coronavirus, it's actually really interesting that there are lots of receptors, multiple different receptors that seem to be able to be used, and serves as a fusion protein. It's a classic trimeric fusion protein. This is also what people will call the class 1 fusion proteins, and again, what Dr. Wiesler's been working on in particular. Some of these viruses have something called a hemagglutinin esterase. So hemagglutinin does what? Agglutinates red blood cells, exactly. So it's another way of the quick and dirty and not terribly high sensitivity assay for looking at virus infections. So, but it turns out not all coronaviruses have these hemagglutinin esterase proteins. Probably the other important protein to talk about, because we haven't really talked too much about the structure of envelope viruses. Yes, flaviviruses, but all that boring disease stuff. Here is the M or matrix protein. And so this is something that is extremely common in enveloped viruses. It's a protein that basically sits between your envelope proteins and the nucleocapsid. It's sort of providing this bridge between the two. So it turns out to be really important for assembly, getting this whole process together. You've got to get your nucleic acid inside the bag of the virion, and then have all of your surface proteins, your envelope proteins that are associated with it. In the case of the M protein, it's incredibly abundant. It's actually the most common protein that you find in these virions. And we'll see why that's important in just a second. So you can make a lot more of this than you have a lot of the other ones. And it bridges across the envelope, interacts with both the N protein, which is the nucleocapsid protein, and the spike protein. So it's really, again, providing this sort of bridge between the two. And we'll see lots of examples of M proteins, matrix proteins, as we continue to talk about envelope viruses through the rest of the course. And then just for so the completeness here, there's also a final E protein. Oh, E reminds me from the flavivirus talk that we had on Wednesday. Dr. Hirsch talked quite a bit about the E protein. That's the envelope protein. So it's the protein that's sitting in the envelope, and that's the one which is also the receptor interacting protein. So the E protein, we looked at some of those dimers as well. In this case, the E protein mostly seems to be involved in virus production rather than interacting with receptors. So what are those receptors? I mentioned this right at the beginning in our outline. A lot of the receptors for coronaviruses seem to be extracellular proteases. And quite why that is a bit of an open question, but it might have to do with the conformational change that has to happen in order to get virus fusion with the host. So these are envelope viruses. They've got a membrane on the outside that has to fuse with some cellular membrane in order to release the virus genome inside the host. So there are, I didn't mention this too far, if you look at sequences of these different coronaviruses, there are two major groups. One of them is the one that doesn't cause the nasty disease, and group 2, this is the one that includes SARS, etc. So there's been a lot of work on this one, a little bit less work on this particular group here. But they do seem to have this commonality that there are peptidases which are being used as receptors. Another thing that Dr. Wiesler pointed out yesterday quite well is that these are, particularly the group 2 coronaviruses, interesting is they don't use just a single receptor. It turns out they can use multiple different receptors. And that's just to get your binding of the spike protein to the host cell. And that seems to allow these viruses to be able to get into lots of different cell types. And potentially also why antivirals and some of the vaccines haven't worked terribly well, because you could block interaction with one receptor, but that doesn't block interactions with other receptors as well. So these are just a number of different ones. Sialic acid has this question mark at the end here. Sialic acid is really important for flu interactions with hosts. So what's doing this interaction? This is a slide from Wiesler's talk from yesterday, which I pulled up from their paper. This is the high-resolution structure of the spike protein. At the end here, now looking down at the top of that, again, these are all trimeric proteins, 3 individual monomers that come together. This top view, this is what interacts with the receptors, lots of different receptors, and it turns out slightly different parts of these proteins up at the top here. Once you have interaction with the receptor, this whole piece basically pops off, and then you have a fusion peptide, and hopefully you remember from looking back at those entry slides when we had all those videos, the fusion peptide is a very hydrophobic part of this protein down here at the bottom, which is normally completely blocked by this other structure on the outside. But as soon as this is removed, that fusion peptide can literally pop out, kind of like a, I don't know, Swiss Army knife or a Leatherman, pulling that particular fusion peptide out, and then can fuse with the cellular membrane to give you this fusion of the two membranes, the envelope of the virus, the membrane of the host, and release the genome from the inside. So there's this process here where this part has to be removed in order to get that conformational change, get to the second in. But the second thing that has to happen is that in order to have this fusion peptide available to make that big conformational change, there has to be a proteolytic cleavage that takes place in the fusion protein right here, which is all part of this spike protein in the coronaviruses, different for different other viruses. You have to have proteolysis, which allows that end to then actually move. Yeah, your question, Ian.
Is this similar to the structure of the influenza fusion proteins?
So the question is this similar to the structure of the influenza fusion proteins? in the overall sense that you have a shorter alpha helix that then becomes a lot longer, yes. There are some slight differences between coronaviruses and some of these other viruses. But the basic idea, where you go from a compact structure to a long alpha helix that has hydrophobic end on it, that's extremely similar with flu, with coronaviruses, and we'll see for a lot of the other viruses as we keep talking about these different things. But that process, conformational change and proteolysis, which then allows the conformational change to take place, are really critical. Okay, so how do you get this? How do you get these? Oh, sorry, Angela.
Oh, well, I was just curious, like, the receptor sort of choice or whatever have anything to do with what's going to happen to it once it gets inside the cytoplasm? Because, like, it has to get to the ER, right?
Okay, so to reproduce, it has to get to the ER, but that's actually after we're getting replication of the virus. But in order to get in, basically, as soon as you get membrane fusion, it doesn't really matter where that membrane fusion is because you've released the genome. And once that genome is released, it gets translated, and then you start to make more. So you can have lots of them. Many of these fusion peptides, they don't seem to be particularly specific. It's just hydrophobic. So once that fusion peptide is released, it'll find any host cell membrane and try and fuse the virus envelope with that host cell membrane. So it's that exposure of the fusion peptide which is the real critical aspect of getting that fusion to take place. So what does the genome look like? Again, these are the largest single-stranded RNA, and this 31,686 is from the beluga whale. So that's where that one is actually from. But it's amazing, 31,000 bases of an RNA genome. This genome, when it comes inside the cell, has a 5' cap, a 3' tail. So looks just like a regular messenger RNA, except most of your regular messenger RNAs aren't 32,000 bases in length. And the process here is also polyproteins. So again, extremely similar to what you saw, or what we saw when we talked about those picoRNA viruses, way back when, polio. So these are all made as one big protein that then gets chopped into smaller pieces. That's mostly true. Turns out that there are two separate sets here, gene 1 and genes 2 through 7. Gene 1 is made as a polyprotein. So this whole process, excuse me here, go forward. This whole protein here is made from this cap, excuse me, getting translated through all of these proteins. Then there's this second gene. This is made through frame shifting that we'll get back to and talk about in just a second. Made as a polyprotein, has to get chopped up by proteases in order to make each of these individual proteins. That chopping up via proteases is done with both cellular proteases and viral proteases. So let's take a quick look at what those are. All of these NS, et cetera, proteins. I'm not going to expect you to remember all of them. I can't remember all the names here. But what's important are really the functions of these that I have circled here. One of them is here, NSP3. NS just stands for non-structural protein. So when we go through a lot of these other viruses, you'll see lots of NSs. Non-structural means that these are the enzymes which are important for making more of the virus. Here, these are proteases that are important for chopping up that polyprotein. Here, second protease. There are multiple proteases that are involved in processing that polyprotein. RNA-dependent RNA polymerase, not surprising. You have to have an RNA-dependent RNA polymerase to make the rest of your genome. And then this, again, also has a cap and a tail to it. How do you make caps? This is made through viral proteins. So we talked about this when we talked about the plant viruses. These are viral proteins that will methylate, they will take the guanine, flip it over, attach it in its weird way. So all of these enzymes here are part of the process in terms of making more of the genome. So that's how you have NS proteins. This whole section is extremely similar to the picoRNA viruses and the flaviviruses. Made as a polyprotein, gets chopped up by viral proteases and also cellular proteases. But then we've got all these other genes down here at the bottom. Spike protein, hemagglutinin esterase, envelope membrane, nucleocapsid, etc. How do those get made? Well, we already talked, sorry, get a little ahead of myself here. So this polyprotein, turns out that the first half, 2/3 of it, is made by normal translation, and this is always being made. And then, in a few cases, and not in all of them, the ribosome, instead of falling off, stays on the RNA and moves to a different reading frame and continues to translate. And this, as we'll see for other viruses, is actually not unusual. We already talked about it for MS2. We talked about the RNA phage. To get the lysis protein, your ribosome just stays on the RNA and moves back and forth a little bit. Here, there's a secondary structure which forms in the RNA called a pseudoknot, which is a base paired-like structure which forms literally right here between the coding sequences for the first part and the second part of these non-structural proteins. Not surprisingly, you end up with fewer of these non-structural proteins than you do of these non-structural proteins. These non-structural proteins include all of those proteases. The polymerases and those modifying enzymes are all in the second part here, where you end up with less of them. So now, what about these other genes here? Now, these are all of our structural proteins. So relative to the non-structural proteins, what do we need for these guys? We need a lot more of all of these proteins relative to all of these non-structural proteins which are involved in making more of the genome. But what do you have to do first? So we talked about one-step growth curves in the last midterm. You need to replicate some more of your genome before you make your structural proteins. How does that replication take place? This should sound extremely similar to what we had a clicker question about earlier. Cytoplasmic vesicles is a really nice EM in the chapter in the textbook we're going to talk about. How do you make these cytoplasmic vesicles? It might be a process like autophagy, so eating yourself. So if you have a coronavirus infected cell, turns out that there are way more intracellular vesicles in those cells than you would normally see in the absence of a coronavirus infection. A lot of those vesicles seem to be that the cell is actually pulling in some of the other membrane components and accumulating those in the cytoplasm, which looks a lot like this autophagy process of eating themselves. So that seems to be a pretty close parallel there. Negative strand, again, this is a positive strand genome. In order to make that genome, you have to make a negative strand, and then from that negative strand, make more of the positive strand. So you have to do this replication process, making double-stranded RNA, again, associated with vesicles. Yeah, Ian?
So is it thought that if it forms these vesicles through autophagy, does the virus induce the autophagy? Is that what's thought to happen, or is it just basically taking advantage of the cell recycling organelles itself?
It does actually seem, the question is basically, is the virus inducing autophagy, or is it just taking advantage of the fact that autophagy is happening? There does seem to be an induction of autophagy, but this is, again, there's a question mark here. It may be an induction, it may not be. But one of the things that you do see is a vast accumulation of vesicles inside the cell, which looks like autophagy. And so, exactly differentiating those aspects is not entirely clear. So we've made negative strand. Now we need to make positive strand, but we also still need to make all of these messenger RNAs for the structural proteins. And if you just isolate messenger RNA from coronavirus-infected cells, what you see is some full-length genome, but a whole bunch of these shorter RNAs, which are, not surprisingly, coding for all of the structural proteins. There are a couple of things that people noticed when they isolated, again, these RNAs from infected cells. One is that all of these RNAs had caps on them, which is not terribly surprising because it's got all the capping machinery. All the non-structural proteins are involved in making caps. But there's also this black box. What's the black box? The black box is a sequence right next to the cap, which is present on this full-length RNA, but also on all these shorter RNAs. But this is not coded for in all of these other sequences. So the question is, what's happening here? How are you getting all of these shorter RNAs? And the answer basically is that the RNA polymerase is non-processive. And non-processive, everyone remembers from that course last term or whenever else you took molecular biology, means that the polymerase pops on and off of its template. And so what happens in this case is you have your polymerase, which will start here at the 3' end of the genome, because all polymerases are always going to go from 5' to 3'. So this polymerase is happily replicating along, and then it'll hit one of these sequences where it turns out that the polymerase actually slows down at that point. And then, because it's not terribly processive, it'll fall off, and in many cases reassociate down here right next to the 5' end of your genome. Makes many copies of these negative strands that are now much shorter than the whole genome itself. These negative strands can now get replicated by the RNA-dependent RNA polymerase to make many copies of these shorter RNAs. And basically what this means is, every time you get to one of these, called them TRS sequences, the name is not important, the sequence is not important. Basically, it's just where the polymerase slows down. So polymerase slows down and has a chance to jump to either the opposite end here, or probably more likely other genomes, which are also present at these cytoplasmic vesicles, and then replicate its way to the other end of the genome. You end up with multiple negative strands, which are present here, that can then be made into the messenger RNAs. So you have many, many copies of these messenger RNAs for the structural proteins, and all of those then get translated into the structural proteins. So here, also known as these nested RNAs, because each of these RNAs is a shorter RNA relative to the whole genome. And it seems that whole genome gets made just because in a few cases you don't actually completely switch genomes. And that's enough of your positive strand RNA to allow enough genomes to be packaged, but you need way more of these structural proteins in order to make a complete virion. So here, making these subgenomic RNAs is the way to get lots of protein. In other cases we've talked about, you get more of these proteins because of translational regulation. Here, it's a transcriptional regulation in terms of how much of the RNAs are being made. So once you've made all of these things, now you need to put it together. This is getting back to where these virions are made. These virions are made at internal membrane structures. Gee, sounds probably vaguely familiar as well. And it turns out that these are almost always then made through budding into either ER or Golgi membranes. And so you have budding into one of these intracellular membrane-bound organelles, then the fusion of this membrane-bound organelle to the plasma membrane, which releases these particles. So if you just look at a coronavirus-infected cell that's producing virions, you'll see all of these enveloped virions inside the ER, inside the Golgi, which then finally get released later on. So this would also be a case of where your eclipse period and your latent period are different when you're doing a one-step growth curve, because these virions here in the intracellular compartment are infectious. All they need to do is get out of the cell. But that process of getting out of the cell also takes time. So we have infectious virion here, no infectious virion outside the cell. Only once you have this fusion did you have release on the outside of the cell. So that's the process here. I'm actually going to skip over this recombinant coronavirus stuff just to give you a really quick overview again of what we talked about so far. The key here with the replication and messenger RNA is that nested subgenomic process. Origins, again, these are reservoir species. The structure has these crowns around the outside, binding to multiple different, almost all of them peptidase receptors on the outside. Translation is pretty normal, releases by budding into the cellular membrane. And just so everybody else has a chance to get 100% on one more clicker question, we'll finish with that. Like flaviviruses, coronaviruses assemble as pentamers at the plasma membrane, the nuclear membrane, at internal membranes. And once you've voted, you can leave. And hopefully it will stop raining for the rest of the weekend. It was so nice for the last couple of days. Yeah?
It's not a question related